It's a pleasure to welcome James Kitchens to give his post-quals talk to CPB. So James joined population biology in 2021 after an undergrad at Warren Wilson College. And during that undergrad and after that undergrad, James had a really interesting set of internships, including one at NASA doing remote sensing of groundwater in California. And so he has a very broad background and a set of quantitative skills, which gives him a broad appreciation of how to visualize data. And James has brought those skills to our lab and to PopBio, and is doing a really nice set of work on visualizing human genetic variation and trying to understand the spatial distribution of genetic variation in humans, which is a key problem both in terms of research and in terms of how we teach genetics at the undergrad level. So it's a pleasure to have James in the lab, and I'm really looking forward to his talk. Thanks, James.
Thank you, Graham, for that introduction, and thanks for coming to my CPB seminar. As Graham kind of said and summed up my talk quite nicely, I'm going to be talking about geographic patterns of genetic variation that we might see in population genetic data sets, how we can use these patterns to investigate questions about our past, and then ultimately ways that we can visualize these patterns to effectively communicate research within our field. And so my talk's going to be broken up into across 3 chapters. The first 2 chapters are going to focus on spatial ancestral recombination graphs, so kind of defining what those are, and using them to reconstruct the locations of genetic ancestors for a recombining sequence, and how might we addressing how we might apply these methods to real genetic data sets. And then the last chapter, Graham and I have developed a new visualization method for specifically studying human genetic diversity and kind of understanding visualization choice and how these choices might affect students' understandings of concepts. And so why do we care about space in the first place? Evolution is inherently a spatial process. If we take individuals sampled from around the world and we kind of look at their genetic similarity, individuals that are closer together are often more genetically similar to one another due to limited dispersal, and this is a phenomenon called isolation by distance. We might also see local adaptation, where environmental conditions may vary across a species range, and the optimum phenotypes or characteristics that are best in those environments may be different. Without the spatial component of these, we won't necessarily be able to understand the underlying genetic diversity that we would observe in these populations. And also a more kind of personal component to this. As humans, we're interested in where our ancestors have come from, how they've moved across our landscape, how closely related we are to one another and individuals are. And this forces us to first think about what we mean when we talk about ancestors. We can think about our genealogical ancestors, and these are all the ancestors that appear above us in our family tree. So our parents, our grandparents, and so on and so forth. And the number of genealogical ancestors that we have grows really rapidly as we look back in the past. So we have 2 parents, they have 2 parents, and so on and so forth. And it not only grows in number, but it grows in spread as well across the world. So these kind of colored bands around, those are representing the genealogical ancestors of these samples in this cartoon. Very rapidly, our genealogical ancestors spread around the world. But in population genetics, we're particularly interested in the ancestors that have passed down their genetic information to us. So we term these the genetic ancestors. They're that subset of lucky individuals that have kind of passed on down their chromosomes to us. And we can look at in our chromosomes, each chromosome is a mosaic of all of these different lineages that have been brought together by recombination. In this case, this is just an image from 23andMe, a 23andMe blog post, which uses chromosome painting, where they're painting different sections of the chromosome based on local ancestry inference, where these particular regions of the chromosome are thought to have come from. And we can see that within our chromosomes, there's many different histories that each one stores, all of which have been brought together, like I said, through recombination. But what's kind of missing from an image like this is an aspect of time. And time is really important when thinking about where our ancestors come from, because if I say I'm from France or Germany or something like that, I'm talking about a specific point in time in the past that I'm referring to my ancestors. Eventually, my ancestors worked their way back to Africa. And in this way, our labels of ancestors are constantly in flux. And Graham and I were particularly interested in how we can track the movement of our ancestors as we go back in time over the generations. And so where this starts is thinking about a pedigree. So this is a pedigree of a sexually reproducing species. And we've marked females with circles, males with squares. And this is particularly a diploid species. So each individual has two haploid chromosomes in this case. And if we have our two individuals at the present day, and we're interested in studying the relatedness between these individuals, we can take each one of their chromosomes and track it back through the pedigree. Ultimately, these chromosomes find a most recent common ancestor altogether at the top of the pedigree at node X. And in this case, I'm tracking just the yellow portion of the chromosome. And this will be important as we're going to go forward. So just this top portion. As we track backwards in time, when two samples meet at the same ancestral chromosome, we call that a coalescent event. So in this case, the coalescent events are marked by K, R, and X. That's just noting that. And at this particular locus and at all individual loci, the relationships between these samples forms a tree. And so if I look at a neighboring region, now we're focused on this blue region of the chromosome, I get a very similar tree, but it's not exactly the same. We've lost node K, and it's been replaced now with a different coalescent node at node P. Looking in the blue region, our sample chromosomes C and D are now more closely related to one another, whereas in our previous example, B and C are the most closely related. So we get very correlated trees when we look at different regions of this chromosome, but potentially slightly different ones. And lastly, just another region of the chromosome. This is just a green region. We once again get a similar but slightly different tree. Looking at all of the trees combined, now we're starting to get a bit of a mess in terms of all of the edges crossing. This can lead to some edge crosses and things like that. But we see that some of the edges are only found in one tree. Some of the edges are found across all of the trees. And it's these recombination events, like marked here, where two separate chromosomes exchange pieces, and now they're brought together onto the same chromosome. And this is represented in kind of a graphical form or a stylized form, where the blue and orange lines come in from the left, the green line comes in from the right, they connect at node Q, and that is then inherited down to the sample, to sample A. And this is what forms the basis of the ancestral recombination graph. So it's a complete description and the most accurate description of the genetic relationships between our samples. It takes all of the individual coalescent trees, weaves them together based on their correlated structure, and then outputs a graph, okay? And this, the graph on the right is going to be kind of our example graph going forward. This is essentially these ancestral recombination graphs can get quite complex, but all of the intuition that we're going to be working with can be worked through on these kind of hand-built simple examples. And importantly, unless we're working with simulated data, where we can directly track the pedigree and the inheritance of these chromosomes over time, the ARG is kind of a theoretical object that we won't know with certainty other than in simulations. So we'll eventually get to. there's been a lot of breakthroughs in ARG inference methods over the last kind of 10 years, taking genetic samples, inferring ARGs to various degrees. And each one of these different methods has some like kind of quirks and things about it. This is a very difficult problem to solve because you're trying to solve a lot of correlated trees, and you have to deal with error and things like that. But there's been a lot of progress in this particular region of the field, and kind of building off this momentum and influx of ARG inference methods, we can start to use these approaches to answer questions about stuff like location inference or ancestral location. So these are two heuristic approaches that have been done in the last few years, where they've taken ARGs and ARG-like structures and estimated the location of ancestors over time, all the way back to the deep past. And so we want to expand on these heuristic approaches and kind of give a more model-based approach that takes into account some of the uncertainty as we go back into the past, and gives a bit more than just a single point estimate for the location of ancestors. And actually, I should point out, this is out of Gideon Bradford's lab, who was a student here not too long ago as well. So it's been cool to chat with him as well about this work. All right. So that brings us into chapter 1, specifically inferring the geographic history of a recombinant lineage using the full ancestral recombination graph. And this chapter has been done in collaboration with Matt Osman and Puneet Dharaj at the University of Toronto. They've been unbelievably helpful working through these methods, and it's been great working with them over the past, I think, 2 years now that we've been kind of head into this. And so to start, it's often easiest to think about this with node recombination. We're just going to work through this problem on a tree and then build the intuition there before jumping into the graph. And so say we know the location of our samples, and we know the relationship between our samples provided by the tree. How might we go about estimating the location of this ancestor, the most recent common ancestor between sample 0 and sample 2, geographically in space? And at least when I look at this problem, I initially think, well, it's probably somewhere between the locations of 0 and 1, or 0 and 2, because they're the most closely related to that ancestor. And then it might also be kind of influenced by the location of sample 1, because it does eventually connect back to sample 1 as well. And so luckily, we can turn to some research or some work from phylogenetic comparative methods to kind of formalize this whole scenario, where we can treat dispersal along each one of these edges as an independent Brownian motion. And each of these sample locations is then a draw from the multivariate normal distribution that's been centered about the root location, wherever that is found in geographic space, and with a variance equal to the dispersal rate, how quickly they're able to move across the landscape, multiplied by this shared time matrix. And the shared time matrix, this is kind of the heart of these methods. What this is looking at is along the diagonal is the amount of time that's occurred since the root for this particular sample. So all of these have 100 generations since the root, since they all make it to the present. And then along the off diagonals, we can look at the amount of shared time that the samples have within the ARG. And so the blue sample and the orange sample, or sample 0 and sample 2, share 50 generations within this tree, because they share this entire branch together. Had a common ancestor and were attached at that point. Whereas they share 0 generations with sample 1. Okay. And in order to locate the ancestor, this node in the past, we can look at how much time this node shares with each one of our samples as well. So in this case, this node shares 50 generations with the blue sample, 50 generations with the orange sample, 0 with the red sample. And this is all we need in order to estimate the location of this node or this genetic ancestor in the past. And so say we want to then apply this same method to an ARG. A slightly more complicated structure. If we want to identify the location of this recombination event, or this recombination node, we need to consider that in this case, there's two paths above it. There's one that takes the orange path around, one that takes the blue path around. And this directly violates one of the assumptions that we had when we started with our tree method, which is that the dispersal along each edge of a tree has to be independent for this method to work, for that method to work. Whereas here, if you go left around the loop versus right around the loop, you get to the same location. And so the dispersal along the left and the dispersal along the right side of the loop have to equal one another. And therefore, they're no longer independent from one another. There's some sort of, we have to take into account that this assumption has now been violated, and we need to turn to a slightly different approach. So instead of focusing on the covariance or the shared time between our samples in the ARG, we can instead calculate the covariance between the paths to the ARG. And so what is a path ultimately? We've got our ARG again, and I've just labeled the nodes so that we can kind of talk about each edge. A path through the ARG goes from one node in the ARG all the way back to the root of the ARG. So in this case, a path from sample node 0 goes through 3 up to 4, 5, and then left around to 7. Okay, that's one path through this ARG. There's 5 paths in total through this ARG, 2 corresponding to sample 0, 2 corresponding to sample 1, 1 corresponding to sample 2. And that's because sample 2 isn't found beneath the recombination node, whereas the other 2 are. So this is an important distinction between ARGs and trees. In a tree, the number of paths through the tree is equal to the number of samples, whereas in ARG, the number of paths is some kind of function of how many samples you have, how many recombination events there are, where those recombination events are within the ARG. And so just as before, we can look at the covariance between our paths, or the shared time between our paths. So in this case, one path from 0 up to 7, another path from 1 up to 7, and look at how many edges they share in common. And so in this case, they share 75 generations within this tree. You can also look at 2 paths from the same sample node. In this case, the 2 paths, one which goes clockwise around the loop, one which goes counterclockwise, they both share the most recent 50 generations, and then they split and go kind of their separate ways. And altogether, we get our shared time matrix between the path through the ARG. And so just like with the trees, this is going to be kind of the heart of our ARG-based method here. So if we want to locate where this recombination node is geographically, we can look at where the samples are, the location of the samples, and we can calculate the covariance between this node, or this path, I should say, this path for this node, and all of the shared paths that we included in our shared time matrix. All right. And the last little bit before getting into some simulation results is we might not want to consider the entire chromosome at the same time, or run the entire chromosome through our analysis. One, that might be completely impossible in terms of computational power. Or ultimately, it might not be possible because there's just too many trees, too many regions. And so we were interested in looking into what might happen if we broke it up into chunks. So here, if I'm interested in this particular region of the chromosome and what's going on there, I can set my window around this region, and I can look at some number of neighboring trees on either side of the region. So in this case, if I have a window of size 0, or marked with W0, then I'm only including the local tree relating to that one region. a window size 1 expands it out to one neighbor on either side of my local tree. Window 2, so on and so forth. And so we can look at how this windowing affects our results. So we ran some simulations and estimated, and then at the end of those simulations, it output an ARG. And we could use then that ARG to estimate the locations of random ancestors within the ARG. So random points along branches, and we can compare our estimates to the true locations of where those ancestors were within our simulations. And what we see is that using the full chromosome in our analysis actually gives us, on average, more error or higher error than if we use smaller windows. And this was a bit puzzling to think about when we first ran into it. Why would giving more information to our system actually give us worse results? And it was a bit discouraging, I think, when I first looked at it. On the other side, the uncertainty or the variance in our estimates greatly decreases by providing more information. So there is some plus side to this. But we started to look at, like, why is this occurring? What's causing the error to increase? And so in this image, we can look at the bias in our location error, where error here is measured as the true value minus the estimated value. And ideally, we wouldn't see a relationship between our error and the true location of the individual. If they're on the left side of the map, we want to be equally as good at predicting their location as if they were on the right side of the map. And so to highlight some specific regions, we see a kind of more strict pattern going on here. These blue regions are the region marked in blue. This is the region where we're estimating the ancestors to be too close to the center of our map compared to what they actually are. Alternatively, this area marked in red, this would be where we're estimating them to be too far away from the center compared to where they actually are. And so what we're seeing is we're estimating a lot of our ancestors, particularly the older ancestors, to be too central compared to what they actually are. And we think that this is after kind of investigating this further, this is due to an incompatibility between Brownian motion and the ARG object that we have. If you have a Brownian particle and it splits into two particles, the likelihood that they come back together and come back into contact with one another is extremely small. But that's exactly what we're saying is happening when we have an ARG. We're saying that they split at some time, they go around either side of a loop, and then they come back together. And so in order for that to actually occur, it means that they must not have gotten very far away from one another in the past when going around the loop. And so what's happening here is when we're including lots of loops in our ARG, it's pulling everyone really close together in the center to make it even possible for them to go about kind of splitting and coming back together and splitting again and coming back together. So this is due to kind of an incompatibility between our model and the object that we're running with it. And it has some kind of interesting quirks with dispersal rate estimates and things like that, that if you're interested, we can kind of get into more. I'm not going to talk about today. And so sticking with another simulation, we can look at this admixture simulation where we start with two populations that are geographically isolated from one another on our map. We allow them to disperse into one another over the course of about 1,000 generations. And then we sample a bunch of individuals from the modern day population. And we can see a clear geographic climb in ancestry where individuals on the left are more blue related, individuals on the right are more orange related in this case. And if we highlight just one single one single sample, this is what their chromosome looks like, where I've colored specific regions of the chromosome according to which subpopulation they came from. And we can track these specific regions over time. So these are the true locations of these two neighboring regions. So over time, at the start of the simulation, they start in their two subpopulations. Over time, they disperse into one another until they hit a recombination event, which brings them onto the same chromosome. From that point on, they're together until the present. And using the windowed approach of version of our method, we can get a very similar picture with our estimates. So the white line is the maximum likelihood estimate of each lineage, and then the colored bands are our confidence intervals around those estimates. So we see a similar pattern where they start off separate deep in the past, come together about 400 generations, and then are stuck together until the present. And so I think that this is one of the areas where our method would do particularly well in being able to track this compared to a tree, which wouldn't have the Y-shaped pattern. They would split off immediately. And so some limitations in all location-based kind of inference, uncertainty in location grows really rapidly as we look deeper into the past. And so these methods are probably best suited for estimating locations of recent ancestors. The geographic distribution of your samples will affect your estimates. You're not going to be likely to estimate that ancestors came from another location unless you have samples from over that in that direction. And then lastly, Brownian motion is an approximation and doesn't consider things like topography, resistance to movement. These individuals will gladly jump over oceans and things like that. And depending on your system, that might be okay. Some systems that would be very, very unlikely. All right. And so thinking about how we could potentially apply these methods to genetic data sets, as we said, there's a bunch of different inference methods that have come out. Only one of these inference methods gives us the exact object that we would use as input, and that's this one at the top, Argweaver. Unfortunately, Argweaver is pretty limited in the number of samples that it can run, and so that would greatly reduce our resolution in terms of our spatial resolution in terms of what we'd be able to infer. So for now, this got us really thinking about, maybe putting aside Brownian motion and thinking about methods that could really capture a lot of the strengths from these other inference methods and do a faster, more efficient, and maybe rougher estimate for location. And that brings us into chapter 2. And in this chapter, we're like I said, we're kind of working with a different movement model. Here, there it goes. Here, we're chopping up space into really small hexagons, and we're going to use a discrete time Markov chain, at least initially, to think about how an individual in one hexagon can move to any of the other hexagons. So if I have a sample that's starting in the central hexagon, it's able to move to any of its neighboring hexagons or remain in the same original hexagon with each transition. So after one time step, in this case, it could be in any one of these hexagons. And there might be different probabilities of transferring to each. But what if we knew a bit more information? What if we knew that there were 2 samples and we knew that these samples coalesced or had a most recent common ancestor 1 generation in the past? Now we're again kind of focusing on trees for the moment rather than arcs. So if these 2 individuals coalesced 1 generation in the past, we can look at all of the possible cells that the orange sample could make it to, all of the possible cells that the blue sample could make it to. And it's the overlap of these that is where this particular ancestor could possibly be, because both of them have to get there. It's only this one cell that it's possible for that ancestor to be in. And so we can take this method and kind of work our way up the tree, including more and more samples. And as a proof of concept, we used Wands et al. Data set. This is actually the same data set that had the GIF earlier in the presentation. Each one of these circles is a geolocated human genetic sample. And so I took land masses, chopped them up into the small hexagon cells, removed any of the hexagons that don't directly connect to the main landmass, just for simplicity's sake. And identified all of the neighbors of each one of these cells, so all of the possible transitions that they could make it to. And then I took just a subset of samples from the original data set and figured out which of the hexagons those samples would necessarily fall into. So here, each one of the orange dots is a group of samples, with the larger dot being more samples found in that particular hexagon. I then applied this to just a single tree, a single random tree on chromosome 17 that was inferred from the WANS data set. And so I ran it all the way up to the root, and this is looking at the most recent common ancestor of all of the samples in this data set. So essentially the root of the tree, what's the most likely location, or excuse me, what's the location of this particular ancestor, which occurred 13,000 generations ago in the past. And so a single tree gives a pretty noisy and kind of low signal estimate, but even still, we can see that the cells corresponding to Africa and broadly in Europe have a higher probability of being the location of that particular ancestor than the cells over in Asia and Australia. And so we have just kind of started this method, but we're thinking about ways that we could combine the signal from a bunch of different trees together, ultimately to give us a clearer picture of, and maybe more fine detail picture of what's going on. I was currently, for that example, just use a transition matrix where you're equally likely to transition to any of your neighbors. But there's much more complex ways that you could think about this. Maybe you're more likely to go through different terrain types and things like that. Or even that your transition rates might change over time. Maybe the conditions over time are changing. So incorporating those two aspects into the model might be another direction that we could take this. But we're also definitely interested in feedback as we're kind of working through the beginnings of this method. All right, I'm going to take a sip of water. All right, and so the last chapter, here, Graham and I were looking at a few different ways that we could visualize and talk about human genetic diversity to understand how students are grappling with these concepts. Ultimately, I'm really interested in visualization methods. They're really fun. And I think it's an interesting and difficult problem because we're taking this really high dimensional complex data set and necessarily having to reduce it down to something that's two-dimensional, maybe three-dimensional. Every visualization method has its strengths and has its weaknesses. It's going to highlight specific components about the data or ignore specific components about the data. And I think that there's some interesting decision-making that goes on that I think is worth considering when developing figures. And so for this particular project, some background, Lewontin came out with this paper, The Apportionment of Human Diversity in the 70s, which looked at how the vast majority of genetic variation is found within human groups rather than between human groups. And this goes to support that race being a social construct rather than a biological construct. And since then, there's been continuous discussion, or before then and since then, there's been continuous discussion about the interplay between genetics and race and how we go about kind of talking about these things. Donovan et al., 2019 on the right, looked into how current biology and genetics curriculum in schools might inadvertently be biasing students towards a more essentialist view of race and the differences between people. And this is an important concept because they found that individuals that have a more essentialist view of race are less likely to support programs that seek to address racial inequalities in societies. Taking this even a step further, genetics research has been repeatedly co-opted by extremist ideologies and misused and misinterpreted to support these extreme beliefs and actions. And I think that this is a really prominent issue with scientific figures in particular, because these figures are very easily passed around social media and can be removed from the context of the original article. And you ultimately don't have much control once it's out there. Carlson et al., which includes our very own Brenna Henn, looked at how figures were being re-annotated and altered, and essentially having their own kind of evolution of these figures over time as they're being passed through different channels. And ultimately, as I said, these figures can have kind of this life beyond their original article. And I think that this is an important thing to think critically about as we're considering developing figures and all of the possible misinterpretations of them. And one such common analysis that's done with human genetic variation, or just genetic variation in general, is principal component analysis, which I'm not saying is a bad analysis by any means. I think it's a really powerful analysis. But I think that it can be difficult sometimes to teach what's going on there and understand. So this is a PCA from Lewis et al. And one interesting thing that they did was they first put down colored dots from a reference panel of 4,000 individuals, and then underlaid a bunch of gray dots that represent 30,000 individuals broadly from New York City. This is from the Biomedas. And so what I think is interesting with this figure is if you just look at the colored dots, you might get the impression that it's a very kind of distinct clusters, or there's a very distinct or discrete landscape to the genetic variation that's going on. But by including the gray dots underneath, you see that there's actually quite continuous structure going on. Between in humans. And I think that this points to, one, that sampling can have a really important aspect, or sampling can have a really important impact on our visualizations and on our interpretations, as well as we don't necessarily know the magnitude of variation difference or magnitude of differentiation between samples on one side of the PCA and samples on the other. Often they are presented with an axis of variation, or excuse me, the percent variation explained along the axes, and those are important. But even still, the visualization itself really likes to pull things apart, as it's good at doing and supposed to do. But that might skew our view of what's actually going on, or the absolute value of differentiation that we see. And so Graham and I were interested in developing a visualization method that looked at kind of the sharing of genetic variation between samples. And here we're looking at data from the 1,000 Genomes Project that was originally processed by Budanda et al. And we've revisualized here. So focusing just on samples that have been identified from being from the Americas, we can look at the common genetic variants present in each one of these samples. So the size of the circle represents the number of common genetic variation, or excuse me, number of common genetic variants within that sample, where common here is being defined as greater than 5% minor allele frequency. And we're interested in the common genetic variants because we think that the common genetic variants better define the group compared to the rare genetic variants, which more define individuals within the group or families within the group, things like that. So that's why we put this cutoff of greater than 5% frequency. So we can see that the sizes of the circles are slightly different from one another just off the bat. But we can also look at the overlap in common genetic variation between our samples. And so on the right, we presented an Euler diagram, or essentially a area proportional Venn diagram, where the amount of area of overlap corresponds to the number of variants that are shared between those specific groups. So in the center of the diagram, those are variants that are common in all of the groups. Towards the edges, those are variants that in this analysis are unique to that particular sample. And we're interested in kind of bringing this into EVE 100, or at least into classrooms broadly. And so we developed an exercise around this to be able to allow students to kind of play around with these overlaps. And so we can have students go to this particular website. It presents them with a sandbox that has, in this case, 3 circles that are sized proportionally to the sample that they represent. We chose just 3 samples rather than 7, just to kind of simplify things so that they're not stuck just playing with circles all day. And so we can present this diagram and allow the students to kind of estimate how much shared variation they would expect to see between these different samples. They can arrange the circles into based on the overlap that they would estimate, and then submit their diagram. And this is submitted anonymously to a Google Sheet, essentially. And what this allows us to do is quickly survey what students' kind of understandings of these topics are, either before we give a lecture on human genetic variation, or after we give a lecture, kind of to see where they're coming, how much they understood. And we can measure statistics like, okay, what's the area that they predicted is common in all of the samples? What is the area that they predicted is unique to all the samples? And use these, compare their estimates against what we actually see in the data set. And so, like I said, we're able to quickly survey a bunch of students in this way. They can participate just on their phone or on a computer. It's an individual activity as well. It's not a discussion that they necessarily have to participate in kind of vocally, which I know a lot of students might be concerned about saying the wrong things. And we can pair this, like I said, with kind of curriculum around these issues, as well as I think it's important to pair this alongside an exercise that makes sure that their intention matches the figure that they submitted. So if they wanted to overlap the circles 50%, but they submitted something that was closer to 25%, that wouldn't be a great activity. We wouldn't be able to use necessarily that data very well. So making sure that they're able to accurately kind of overlap the circles, how much they want to overlap the circles. And so, yeah, we're hopeful to potentially implement this in a class like EVE 100. We ran through a just brief example of this during one of the discussion sections, not to collect any data or anything, but just to make sure that the activity ran properly, and it seemed like they engaged with it. And so, yeah, excited to kind of continue building off of this in the future. And with that, I'd like to acknowledge kind of everyone that's helped me through this project, the COOP lab and collaborators, as well as the 3rd-year cohort who've been great, population biology graduate group as a whole, and CPB funding source, and then my wife and my parents for listening to many practice talks. But yeah, thank you. I think I'm running questions, so if anyone has questions.
For the spatial arcs, you said that recent is okay, and if you get deeper in the past, the uncertainty. Can you give us some kind of intuition for what deep past is? How to think about it in terms of population size and time?
Yeah. For our simulations, we really wanted to cut it off relatively early because we were simulating individuals essentially in a box that had reflecting boundaries, which wouldn't behave like Brownian motion. So essentially, anytime you're going to start running into boundaries in your dispersal, which is often, you're going to start having behavior that isn't necessarily Brownian and could run into issues. And so it would be kind of system dependent on how quickly your environment's changing, how quickly, at least in our simulations, like 2000 generations we kind of have to cut it off, but that's quite deep into the past as well, so. And I think that's something we want to investigate for. Yeah, either one, right?
I think your teaching tool is super cool. And you mentioned just a little bit like trying it out in other classes and maybe collecting data. What data would you collect on it?
Yeah, it's a good question. And this is something that Graham and I are kind of be going through IRB to kind of work out. Essentially, we'd be interested in being able to see the distribution of statistics, like how much they're predicting is common, how much they're predicting is unique, and maybe even what essentially the activity does now is it records the positions of the circles that they've placed so that we can calculate and we would even be able to recreate the figure that they developed. That's essentially the information that we'd be interested in to compare the distribution of class expectations to what is actually found within the data set as a whole.
Yeah, very nice. I wondered if you're using this discretized sort of map approach to estimate locations in the past, like would you be able to incorporate sort of known dispersal events, or if there's like an invasive species and for instance it's known that it was brought on such a ship at such a time or something.
Oh, interesting.
Like, is there other information that you could incorporate rather than just kind of assuming this model of bouncing around?
Yeah, that's an interesting concept. And I wonder if there's a way to kind of add that to the graph itself. Like you have the connections between the samples and those should say, oh, potentially suddenly they went from being in one location, but now they're tracking back to a bunch of samples over in another region. Like the hope would be that you could have some sort of connection in that way. In the discrete model, right now, one thing that we run into in the proof of concept was, well, what happens if the samples are far enough apart that with my simple transition matrix, they wouldn't actually be able to get to one another in time. Clearly, I'm missing something about how they moved, maybe in the recent past or distant, I don't know exactly. And so potentially incorporating that somehow into the transition matrix, a bit more nuance into the transition matrix could help solve some of those things, maybe. But yeah, it'd be an interesting direction. Great. Thanks, guys.